uh, just a little bit about where I come from, my experience and obsession with NAD and niacin. When I was a teenager in the 80s, my grandmother had a book called Life Extension by Dirk Pearson and Sandy Shaw, and they were physicists and really highly experimental people from in California that were excited about life extension, and they wrote about niacin and how it caused this flush response, but it it always did amazing things for all of the lipids. And when I got to graduate school, I would take this, and my colleagues would say that can't be good for you because I'd be turning bright red. And then when I got to the but the lipids always got better, you know, when it was measured. And then so when I got to graduate to where I had my own laboratory, and I was in a position that I could really explore this. You know, I need to figure this out. How does this niacin work? And then that was about 2004, and then I learned of Abram Hoffer, and then he taught me about the recent discovery of a third form of vitamin B3 and nicotinamide riboside, and the rest is history. NAD really experienced a real renaissance starting in about 2004. So by the end of this talk, hopefully everyone will appreciate that NAD is sort of the center of the biochemical universe, where it represents a lack of where a deficiency represents a lack of a true molecular part of the human body machine that controls susceptibility to nearly all disease and associated progression. And you will understand that vitamin B3 is the dietary precursor to nicotinamide adenine dinucleotide, or NAD. And you'll understand the unique characteristics and advantages of the different NAD precursors. And for me, that has been the most important question and my obsession for about 15 years. Which NAD precursor is the best, and how should it be administered? And one thing that is just completely not explored is the consideration of glutamine in this whole thing. Glutamine turns out to be really interesting, and also the advantages of niacin as compared to dimethyl fumarate, which is, let me see, I'm trying to see if I can hide it. Okay. And then in the end, we'll have detailed takeaways that you can consider in your practice for specific indications based on clinical research. So the history of NAD is really interesting. The losses due to the NAD deficiency disease pellagra were the greatest losses of any nutritional deficiency in the history of the United States of America. However, it should be noted in the history of nutrition medicine, perhaps scurvy was more devastating. In the 19th century, they had to literally kidnap drunken sailors to get them on the boats, because they knew that once they got on the boats and headed out to that ocean, they'd probably die, and countless people died from scurvy. But in both cases, these nutritional deficiencies speak volumes to the susceptibility of the human body machine to their deficiencies. Classic pellagra NAD deficiency was worldwide in 1942. It was started, but with the invention of the mill in at least in America, where it became accessible that everybody could have white flour and white rice, actually, starting in the 1870s. By the first 3 decades of the 1900s, over 3 million people, or roughly 1 in 33, were affected, and it killed more than 100,000 people. The symptoms of niacin deficiency, which we should consider when we look at patients, are diarrhea, dementia, dermatitis, and ultimately death, and just general categories. And initially there was all of this association with corn, and all these meetings to try to identify some infectious agent, but eventually, they had to figure this out. There was insane asylums full of people with skin problems, just dying, and all kinds of mental health issues. And so the president hired an epidemiologist who eventually noticed that people who had a cow, and they were okay. But people who ate molasses and corn, they suffered. And he discovered that brewer's yeast could cure it. And eventually they discovered the molecule in brewer's yeast. By 1942, they had realized the effects of this were so profound on people recovering from what looked like schizophrenia and just all kinds of suffering, that the government made it a law. And when a government makes it a law to fortify something, think about what it takes to do that. They made it a law that you had to put this in, you had to fortify flour and rice. And to this day, in 2021, we take this for granted. And in my opinion, this would be something to consider for COVID-19 as a mandatory fortification, but that's another subject. By the 1950s, Abram Hoffer really took the ball to another level. He was a biochemist at the time that so yeah, here we have NAD as a the Achilles heel of human susceptibility to disease. And here's where Abram Hoffer did some of the first work with high doses of niacin, realizing that nicotinic acid, which is niacin, a chemistry term for it, even to this day in 2021, it raises high density lipoproteins, HDL, more than any known pharmaceutical, more than all of the statins. The nicotinic acid decreases, at the same time, it decreases VLDL, triglycerides, and it can correct cholesterol up or down, and it usually works when administered properly. And then in about, so the vitamins niacin and niacinamide were discovered in roughly 1939. And then in 2004, a third form was discovered by Charles Brenner, who was at Dartmouth, then was the chair of biochemistry, Iowa, and just moved to City of Hope in Duarte, California. So so this really, this was interesting because it's interesting what happens when you discover a vitamin in 1939 versus 2004. In 1939, you know, the government made it a fortification, but in 2004, it got patented, and now it's sold at literally 100 times higher cost than niacin. But as you'll see, it doesn't appear to be as good as niacin, in my opinion. But it's under investigation. Nicotinamide riboside still. Sorry about the typo, not ribosome. And about 2004, there's a lot of lifespan research, genetics from Leonard Guarante's lab in Harvard and David Sinclair, where they did a lot of work starting in yeast, unbiased genetic screens, and found that sirtuins can mediate replicative lifespan with respect to caloric restriction increases in lifespan, and we'll get to that later. And then just last year, you know, there was a really nice clinical trial in Finland doing three times 1 gram of niacin with profound effects, decreasing liver fatty content by 50% and increasing muscle strength in adult onset mitochondrial myopathy. And you also wonder about the historical implications of niacin. You know, prior to the fortification of niacin, we had, you know, to it really entering the supply, we had these world wars where apparently, you know, several countries were trying to take over the whole world, and just, you know, unspeakable horrors. We still have a lot of things, but it was interesting with these hamsters studies. These ecologists noticed hamsters that were eating their own. They didn't have the maternal instinct to care about themselves or their siblings, and they were eating mostly corn. And the thing about corn is it does have a precursor to NAD, but you have to add lye. You have to add base to extract it. The Native Americans, they used to do the process of nixtamalization, where they'd add lye and get it out, and so then corn would somehow they figured that out. But you really have to ask yourself about how niacin affects this sort of sympathy, maternal instinct, and the greater ramifications for history. So here, Abram Hoffer has carried the torch more than anybody, in my opinion, for what's going on with niacin research, and I try my best to honor his legacy. I had the greatest privilege to know him during his last years, and he was such a great example of generosity scientifically, and his clinical generosity, and he practiced medicine for all the right reasons. And so his story basically was, he was a PhD in biochemistry in the 1940s, observing the Indian reservations in Canada, and trying to recommend how much to fortify, and they weren't always listening to him. So, you know, he was raised on a farm. He had really strong willpower and work ethic. And so he went back to school, became a medical doctor, and then he was put in charge of a very big mental asylum in the middle of Canada, Saskatchewan. And he noticed the schizophrenics in the 1950s. They resembled some of the people suffering from dementia previously. So he asked Merck if he could get some niacin, nicotinic acid. They sent him a whole oil drum full of it. And he thought, you know, maybe these individuals required higher amounts. And he's pretty much proven that. And now we've even shown it genetically. There's a nice review by Bruce Ames that shows some specific examples where the binding affinity for NAD is reduced due to mutations. And so the science is very solid now that there are some people with genetic dependence and likely similar environmental caused greater dependence for higher doses of niacin and respective NAD that's formed in your body. So he treated over 5,000 schizophrenics with high doses of niacin and found that he could cure a lot of them, not as much some of the chronic long-term cases. And he developed the first biochemical theory for schizophrenia, which involves noradrenaline getting converted to something called adrenochrome, where NAD is used to metabolize and clear this. And NAD is very important for metabolism, all xenobiotics and drugs and basic metabolism. It's required for it as a cofactor. He introduced Linus Pauling to high doses of niacin, and Linus learned about vitamin C and how human beings and guinea pigs have a mutation where we do not make more vitamin C when we have an infection, but all the other animals do. For example, a goat will make much more vitamin C when it's infected, but human beings don't. And Linus came up with this term, orthomolecular medicine, in 1968, and the rest is history, where niacin and vitamin C are sort of the two classic high-dose orthomolecular medicines. Ultimately, at the end of his life, he concluded that some people, and this is after treating thousands, need as much as 18 grams of niacin in divided doses over a course of a day, which is shocking. You know, for me to take that amount, it would be terribly unpleasant. For some people, 500 milligrams is a little is unpleasant. But schizophrenics typically do not flush as much. Perhaps the molecule is going into the cell instead of the other receptor-mediated flush pathway much more in these individuals. And there's other possible explanations for that. But in the first, his first clinical trials in the 50s, this is one of the first ones. He used 3 grams of niacinamide or the flush-inducing nicotinic acid, and he got 80% versus 30% recovery rate at 1 year. He noticed that many schizophrenics don't flush until after 2 years of treatment. And it's very curious. And Dr. Horobin, who's in the Orthomolecular Hall of Fame, had previously observed decreased prostaglandin E2, which, by the way, is formed from the precursor linoleic acid. He decreased amounts of PGE2 in schizophrenics, Of course, NAD is surely in this biochemical pathway as well. Anyways, something to keep in mind that some people, they do not flush for a long time until they start to get better. So to flush is a healthy response. He, in one of his first experiments, his first patients this patient, he said the response was so dramatic that he said, once you see this happen where a schizophrenic patient is in a stupor and recovers, you never forget this. And so he did 6 clinical trials up until the 60s. So what could be causing these NAD deficiencies? Well, with pellagra, it was the sudden accessibility of unfortified rice and flour, and and then they figured out how to solve the solution, solve it, and then what could cause it today? Well, there is one of the biggest things is hyperactivation of poly-ADP ribose polymerase, which uses NAD in a polymerization reaction to make poly-ADP ribose, which it has typically like 60 to 300 units. Each one is created by degradation of NAD, and PARP is activated by lots of different stresses, any type of DNA damage, including chemotherapeutics, and high calorie nutrition with malnutrition is a kind of a ironic mechanism where you're eating more, your body wants more, but you're damaging it so much with oxidative stress from sugar, and this can activate PARP. There's still a lot more. There's genetic dependence. There's a NAD binding domain that we can kind of see with consensus primary sequence of the DNA for the genes. And you can see that there's some substitutions sometimes that result in weaker binding of NAD. And so you need higher amounts of NAD for the gene product to function, for the enzyme to function. Tuberculosis deserves so much more focus with respect to NAD deficiency. There's a lot of interesting, very provocative things to consider with tuberculosis. I need to look into it more myself. And then with autoimmune disease, we'll talk about that. Your body can actually make NAD without the vitamin. It can make it from tryptophan, but tryptophan is the lowest concentration amino acid, and it's essential, and it's used for melatonin and serotonin in a separate pathway. And it's very interesting. We'll talk about how tryptophan is used to control autoreactive T cells. NAD levels, we now know that NAD levels are decreased as we age. We now know that every night when we go to sleep, there is a circadian repletion biosynthesis of NAD. And then there's this is more of a sort of a dependence or requirement at the bottom. The whole environmental drug metabolism, xenobiotic metabolism, removal of pollutants, detoxification pathways. You need more NAD when you are exposed to these things. So what can we learn from all of the genetic research? Well, you know, sometimes there's so much genetic research and we sequence the whole genome and you ask yourself, how is this actually useful at a practical level? Well, here you go. We can learn a ton starting from these databases. Um, uh, you can go to my website. I have spreadsheets. I worked with the Unipro protein database and these Swiss bioinformatics people to help me refine my search and identify how many gene products use NAD or NADP or NADPH or NADH. And it's over 450 gene functions, and that's a far higher number than any other vitamin. You can see thiamine, vitamin B1, the first one, is only 21 genes, but they're really important for everything, particularly with sugar metabolism and diabetes and so on. And then riboflavin is many genes, B6 is many genes, and then magnesium is just more than anything. So you know, if you ever had in a biology course, we all learn that NAD is required for making ATP with glycolysis and oxidative phosphorylation. But again, it is so much more than the ATP. With the P450 enzymes, these are the phase 1 drug metabolizing enzymes. There's 55 human genes for these. They're involved in prostaglandin synthesis, drug metabolism, all of the steroids, exposure to xenobiotics, vitamin A, vitamin D, and NAD can be rate limiting in the clearance of pollutants and toxins. And as Abram observed, endogenous toxins like this stress induced adrenochrome molecule, which by the way, they injected that and it caused hallucinations. And this adrenochrome is present in schizophrenics, they showed. So, and basic just biochemical homeostasis. For example, with cholesterol, when you take niacin, it can raise or lower cholesterol. So the general concept is shown in the bottom. You have a substrate, like if you're exposed to dioxin, you have a whole bunch of substrate, and then the cell will make, in the case of dioxin, for example, CYP1A1, and it will respond to this dioxin by making as much as 20% of the protein, the total protein of the cell. It becomes CYP1A1. So at this point, you have the substrate, you have the enzyme, but you just have low levels of NAD. So here is an example where if you take high doses of niacin, you can then increase the rate of the whole reaction. So this general concept is true for countless reactions. I mean, just when you think about all the molecules being metabolized by the P450 enzymes. So this pathway shows just the complexity again. For the mechanism of NAD depletion, you have all these substrates on the left, and then the stimuli causing activation of PARP, which depletes NAD, and as PARP is depleted, and on the left side you can see that if it's too much depletion, then what really happens is all of the ATP gets depleted because there's a mechanism for trying to regenerate that NAD that consumes the ATP, and the cell can just collapse. And then it just burst with necrosis, or it can be more controlled apoptosis if there's not such a strong stimuli. So you can see to the right, the categorical uses of NAD as a cofactor in over 200 reactions, and then as a substrate in the sirtuin reactions, the PARP reactions, which are up to 18 now, but we'll focus on one, and CD38, which is really on the edge still of research in controlling NAD levels. And of course, what we all know about NAD in glycolysis and oxidative phosphorylation. So the important question for me for 15 years has been, and running, what is the best way to keep your NAD levels elevated? And you can see in green all of the things that are used in the clinic. And so you see niacin to the upper left, and it goes through 3 reactions to get to NAD, and it requires glutamine. Tryptophan coming down also requires glutamine. And then you have on the lower left, niacinamide or nicotinic riboside. And then I should also have nicotinamide mononucleotide in green, as these are all used as precursors to NAD. This just shows the vasodilation pathway that's unique to niacin. In my opinion, niacin is still the best niacin, but not niacinamide and not so much nicotinamide riboside to my knowledge. Niacin is the only one that really corrects high triglycerides, high or low cholesterol, and all its lipodystrophy concerns. Niacinamide does not, and statins have many terrible side effects. And statins do not lower LPA either. So the unique feature of niacin, really distinct, the most distinctive feature from these other NAD precursors, is that it goes through this other receptor that binds to general monocarboxylates, like beta alanine, for example. And this receptor is in some in keratinocytes and some antigen presenting cells. And when it binds to it, it produces really high levels of prostaglandins. And these prostaglandins are the ones that really mediate the flush response, the vasodilation, the red skin, the head to toe, your whole body can turn almost red. Some people, it's you know, I've been doing it for 10 years and it's completely safe, but people need to be made aware of it because it can be scary and all that. So then it binds to the smooth muscles and works through this pathway, niacin. And this is important because it also, in my view, provides a unique delivery aspect to this NAD precursor, because now the distal capillary beds, including inside your brain, this receptor is in your brain, and it increases your focus, your brain focus. It increases the, it causes a vasodilation in some otherwise hard to reach places that now, and these capillary beds can receive, perhaps receive better this NAD precursor. So this PGE2 though is also a fascinating molecule. On the left, you see a screen using zebrafish. I was a zebrafish researcher from about the year 2000 until 2008 or something. And zebrafish in the lower left, that's the zebrafish. It's about 1 day old, and it's a transparent vertebrate animal model that is small enough you can put it into a 96 well plate and do small molecule drug screens. You can see that in the lower left. And so at Harvard, Dr. Lin Zhan did this screen for small molecules that will increase stem cells. They could do a stain for the stem cells and do a good screen for a lot of things. And they discovered that PGE2, prostaglandin E2, boosts stem cells. Well, niacin increases PGE2 a lot. And so maybe niacin increases stem cells. This is something that people should look at. And then PGE2 also can be used to make cells tolerogenic, meaning if you have autoreactive T cells, the communicating antigen presenting cells can be made to calm those autoreactive T cells down to make them so that they tolerate the self using PGE2. So this pathway may be important, and I'll show an example where high doses of niacin helped in an autoimmune condition. So here this is another representation of the unique aspect of niacin. You can see in the lower left, niacinamide or nicotinamide riboside going to make NAD, which they all can activate sirtuin, for example, which is really famous for roles in lifespan. But nicotinic acid can also go through this sort of heart healthy pathway up above through a 7-transmembrane GPCR to correct lipodystrophy and ultimately reduce mortality from heart disease. And some clinical trials will go over. But I should emphasize that that monotherapy is not if I like sea salt because it has 80 trace minerals, and we should just think about more than just niacin. This pathway just shows the complexity from it's a lot to think about, and it's not just all pathways are only as good as the full complement of what is required for the pathway. And this shows another whole nother aspect of NAD. With alcoholics, there's a real interesting history. But this shows exactly where NAD is involved in clearing the toxins associated with consuming too much alcohol. And there's lots of case studies and with NAD correcting, curing people with alcohol problems. So here's clinical trials. So this is generally prioritized from the highest quality studies to the lowest available quality studies we currently have. For lipodystrophy, this is the most established. This has been used for over 60 years now with safety. The concern with liver toxicity is slightly more with the timed release version, but generally it's a misleading concept. NAD increases, as I mentioned, the enzyme activities of a lot of things. So it can be misleading, giving elevated liver enzyme activities that are not truly indicative of a problem. And in fact, it reduces liver fat. We'll go over that. And that's just not a concern generally. So this is established that 3 times 1 gram a day of niacin, or it can be less, can correct elevated VLDL, LDL, triglycerides, cholesterol, and can raise HDL more than any statin. And it also can favorably decrease C-reactive protein and influence fibrinogen, which is has a very strong correlation with cardiovascular events. Fibrinogen does. Atherosclerotic lesion regression has reduced carotid atherosclerosis 2 grams a day in clinical trials. And last year in mitochondrial myopathy, we'll go over this, amazing results. And in the 1950s, it was shown that if you, by Klenner, as described by Andrew Soll, that if you take in high frequency 250 milligrams of niacinamide 10 times a day, it can really improve the mobility, increase the strength, and eliminate pain for patients with arthritis. This is something that's not generally, to my knowledge, people still aren't carrying this torch. It's been used in erectile dysfunction studies where it can work. There's examples with alcoholism and depression, multiple examples. This case study was used for lipodystrophy, but it had the side effect of eliminating his desire for alcohol, and the depression went away too. There was an RCT with 1/3 of the patients out of 30 getting better. And in depression, it's been used or niacin or niacinamide in case studies with success. Bo Johnson has done wonderful stuff with eliminating the need for antidepressants. Okay, so if we compare niacin to the most popularly prescribed pharmaceutical in the last 10 years, you see that actually niacin raises HDL more. It lowers LPA where statins don't. The whole statin story has been rather misleading. They use relative risk reduction instead of absolute risk reduction. So where 2 out of 200 events in a high risk population becomes 1 out of 200 events, they describe this as a 50% reduction, but it's actually a 0.5% absolute reduction. It's been very misleading. And then if you look at the cost, niacin is $5 a month or cheaper, and statins typically $50 to $300 a month. Side effects of niacin are rare. There is one to be aware of, it's a macular cyst that does occur sometimes with niacin, and it goes away when they stop taking niacin. So this is something to be very aware of. I don't know if this has become a serious problem or not, but it is something, and we don't know, at least I don't know, if this is caused by the flush or the NAD pathway. And then with statins, there's muscle myopathy from interference with coenzyme Q, and there's memory issues likely from interference with cholesterol metabolism that we'll go over. So nicotinic acid, niacin, has been used for over 60 years now, and you can see how it favorably corrects triglycerides, VLDL, LDL, HDL, and lipoprotein A. What you can see the general direction, even if the progress of clinical medicine feels like it's moving like a glacier, like it does to me at times. The NAD responsive indications remain to be determined. There's so much to be determined. So, for example, we'll go over the curing of age-related mitochondrial myopathy reported last year, and you can, by extension, consider that high-dose niacin may be useful for all of us as we age in addressing age-related sarcopenia, loss of muscle. Autoimmune diseases in general, you have activation of a gene called endolamine 2,3-dioxygenase, or IDO. And this enzyme is fascinating for controlling your synthesis of NAD. We'll get to that, but it's observed in all of the autoimmune diseases and plays a dramatic role, strong role in controlling autoreactivity of the immune system. NAD in acute critical care medicine is still largely unexplored, but I'll show you dramatic effects with strokes. In neurodegenerative diseases, we'll really cover that tomorrow but it's clear that NAD is really important in the most bioenergetically demanding organ of your body, your brain, which is only 2% of the body weight of your body, but it consumes 20% of your energy. So it's real important for the energy component, but also for the trying to maintain protein folding for all of these proteinopathies that occur in neurodegenerative disease, but we'll cover that tomorrow. Kidney disease, there's great data on this now. I won't get there though. And then sleep. Sleep is the most important thing for health, and how we can think about NAD in this context. We won't really get there either, and tuberculosis is another global concern that deserves much more focus. So last year, here's a completed study that is really exciting. You know, it's how often do you see paper anymore where the word cure is used? And it was used last year. In Finland, they treated patients with mitochondrial myopathy diagnosed by biopsy with 3 times 1 gram of niacin for 3 months. And they really measured it well with the assistance of Dr. Charles Brenner. And he was a pioneer in NAD metabolomics. And it increased NAD levels by 7-fold, and even in these patients suffering from the disease, and even 6-fold in the controls. This is a really high increase. And if you look by comparison in some of the higher dose studies with nicotinamide riboside, it was a 1.4-fold increase after 2 weeks by comparison. I should say nicotinamide riboside, and I excuse the typo, it's not ribosome. It continues to be an active area of investigation. It was only an active molecule under study. It was only discovered in 2004, but thus far, and I've done some experiments myself, I'm still more impressed with niacin personally. It decreased the liver fat by 50%. It increased the strength in these patients 10-fold in abdominal in some of the patients, and they measured about 5 different muscle groups. And then, but some patients had a tendency towards anemia. So this, you might pay attention to this. I'm not sure what could be going on there. I don't know if somehow it's related to the methylation pathway. I can't figure that out. But mitochondrial myopathy, the disease presents with weakness of eye muscles, and niacin rescues this. So basic research has also shown recently that NAD supplementation can prevent amyloidosis in muscles. This is so NAD and NAD deficiency has been observed in sarcopenia, the general population. So again, it argues that as we get older, it's probably a good idea for all of us to take niacin or related molecules to address this muscle weakness and loss. This is the paper. Niacin cures systemic NAD deficiency and improves muscle onset, muscle performance in adult onset mitochondrial myopathy. You can see in the upper right the dramatic increase in NAD after taking 3 times 1 gram of niacin. And in the lower right, you can see the increases in muscle strength. Now, glutamine is something that nobody thinks about with respect to NAD. But glutamine is a fascinating amino acid. It's conditionally essential. Some tumors require it as well. And it's used on the pathway on the left to convert nicotinic acid adenine dinucleotide to nicotinamide adenine dinucleotide, NAD. And you may ask, why could it be essential? And I really started to focus on this, I found there's a very rare deficiency called glutamine synthase deficiency. There's only something like 4 patients known. And then I suggested to the geneticists that, maybe nicotinamide could be useful for these patients, because maybe glutamine is most essential in this pathway on the left to making NAD. Maybe its conditionally essential function is in making NAD. And so nicotinamide does not require glutamine. You can see in the lower left, nicotinamide will go on to make NAD without needing the glutamine, which is the GLN. So they tried these experiments in basic cells, and they showed that glutamine itself can increase NAD. And that's not surprising when you think about it, because you look on the figure on the left, tryptophan requires glutamine, so glutamine can be rate limiting. Niacin requires glutamine, glutamine can be rate limiting. And glutamine, as far as clinical trials, it's been shown to be really active in helping burn patients recover, and cancer cachexia, the desire to eat and so on, and wasting away. The other thing was the glutamine synthase deficiency this geneticist was studying. It resembles pellagra, shown in the right. This poor person has what looks like dermatitis all over. Here, this is showing this study from Johannes Haberle where he took the nicotinamide and did the study. And you can see that in a lower panel, glutamine itself can boost NAD levels, even in controls. So it looks like maybe nicotinamide may be very beneficial. So far, I think there's some effects with the patient. There's only like 2 or 3 patients, and they're I don't know how many are alive currently. But as far as lifespan research goes, all roads have been converging on NAD. Caloric restriction is the only pathway that's been shown to only way that has been shown to increase lifespan in all model organisms, and caloric restriction boosts NAD. Genetic screens for, and we'll get, any genetic screens have identified sirtuins, which are NAD, sirtuins use NAD, screen for activators of the sirtuin identified resveratrol, and I've done experiments with resveratrol where I believe, I observed resveratrol actually boosts the synthesis of NAD, and I believe others have seen this too. So I think resveratrol might largely work via increasing steady state levels of NAD, not just activating the sirtuins. But this shows just some genes shown from genetic research that all are in the NAD pathway, the gold colored ones that are in life, have been shown to be important in lifespan. So a big concern is always safety. Niacin's been used for over 60 years at 3 grams a day, 3 times 1 gram a day. But you really have to warn your patients about the flush. It can be very scary to the uninitiated, and you should start low and just get to know the flush. I think it's best to try to have a minimal flush. And schizophrenics often do not flush and take time to flush. And there's even a skin test for detecting this that's marketed to people who work with schizophrenics, because they often have a reduced flush response. Liver toxicity concerns are misunderstood frequently because NAD is increasing activity of enzymes many times because it's used as a cofactor. When considering niacin or niacinamide, niacinamide appears to be more likely to cause nausea, but niacinamide can be used instead of niacin for a lot of things. And yeah, so they're very safe. With multiple sclerosis, there's a drug called dimethyl tecfidera, which is dimethyl fumarate. It's been sold for as much as $5,000 a month and shown to work via the flush pathway that niacin activates, but it doesn't even work as a precursor to NAD biosynthesis. So it would appear niacin is likely to work much better. It turns out the B vitamin relative beta alanine also activates this flush pathway. This just shows a potential use for NAD in critical care medicine. They did a, this is a really interesting, here we go. So in this study, they occluded the cerebrovascular artery of mice, and they administered NAD spraying it into the nose, one of the nostrils. And you can see that when they did this on one side, it could prevent this infarction in the stroke model. The NAD could keep the cells alive, and that's because NAD is required for anaerobic glycolysis. And so NAD might be useful in diseases of acute hypoxia in critical care medicine, you can imagine. Here's the IDO story briefly. When a woman is pregnant, she has a foreign body in her. And so how is it that she doesn't reject this foreign body? Well, surrounding the fetus, there's some cells that make this tryptophan depleting enzyme. And so the lack of tryptophan prevents the T cells from attacking and killing the foreign body. In mice, if they inhibit this enzyme, the fetus is rejected. And this has now been studied by Andrew Meller and David Munn at Medical College of Georgia very thoroughly, and they've shown it looks like a depletion of the essential amino acid tryptophan in the extracellular media that prevents the autoreactive T cells from rejecting the fetus. And what's more, in all of the autoimmune diseases, tryptophan levels are decreased, IDO enzyme levels are increased. And so a lot of these, they actually look like anatomically localized pellagra in a sense, with depletion of this de novo NAD biosynthesis pathway from tryptophan, where NAD supplementation may be very helpful in the context of autoimmune diseases. In this slide at the bottom, there's a book called The Sun is My Enemy, where this patient suffered her whole life from lupus, and she went and got injected with the Bulgarian drug, which was nicotinic acid, and she got much better. Much more research should be done on niacin and NAD precursors in the context of autoimmune diseases. This just shows a price breakdown for the precursors to NAD, as well as the multiple sclerosis oral drug Tecfidera. You can see that niacin or niacinamide, you know, they're basically 70 years old now, and nicotinamide riboside is only 15 years old, and you can see the difference in price. Niacin literally costs about as much as food, and in my experience, and I've done experiments comparing these in zebrafish models and been looking at the research, niacin appears to be the best, in my opinion. So the conclusions are that highest quality studies indicate that for lipodystrophy, you can use, you know, 3 times 1 gram a day, or 500 milligrams 3 times a day. There's really high quality studies showing most people lipodystrophy can be corrected. Not everybody's aware of this, but it's very clear. And then the cardiovascular risk can be reduced, atherosclerosis reduced, mitochondrial myopathy is now known to be addressed with niacin. Non-chronic schizophrenics can be cured. Not all cases, of course, where Abram observed that some of them are basically dependent. They have to always take it. It's not a cure in the sense like antibiotic. And then niacin should be considered for arthritis. And then there's lower quality studies, but it's been shown it can work absolutely for depression, bipolar disorder, alcoholism, anxiety. And then there's just a ton of things to be determined. Glutamine is something that really should be considered as well. You can take very high doses of glutamine, like teaspoons. It's very safe. It's the most abundant amino acid in your serum. Niacin, niacin is expected to be superior to dimethyl fumarate for treating multiple sclerosis, in my opinion. And always we must keep in mind, though, that all pathways are only as robust as the limiting chain in the biochemical pathway. So we should consider all essential molecules. For example, a boiled egg has all of the essential amino acids and everything for life. That's what an egg is. It has everything to create life. Walnuts are an exceptional nut. They have an activator of ghrelin. They have alpha linoleic acid, the essential fatty acid. And there's really good studies showing people who eat cups of nuts have decreased their risk of cognitive dysfunction. And then sea salt should be used, not table salt, because there's still things like, believe it or not, molybdenum has an essential function. There's so many tiny things that we need that we don't. Nickel has functions. but sea salt provides as much as 80 trace minerals. I've heard. And then finally, you always have to consider when treating people that sometimes elimination is more important than any of this. It can be food allergies, addictions, just stresses, and good sleep. It is more important than water in some studies. Good sleep is also more important than anything. They've deprived mice of sleep and deprived them of water, and the ones that were deprived of sleep died before the ones who were deprived of water, according to Dr. Matthew Walker, who's done a great book here. Okay, so just some acknowledgements. My family, my uncle, my mother, my children, my dad in the upper left, and then Abram Hoffer, who was an example of scientific generosity, clinical generosity, just the biggest influence and inspiration to me. Andrew Saul, likewise, just a great example of tactfulness and generosity and connection and the greater good. Doctors Myron Jacobson and Elaine Jacobson, who did the first NAD biosynthesis assays and methods in enzymology in the 70s, and who continue to focus on NAD, including a company called Niacin with skin creams that have superior delivery. Dr. Kirkland, who's done the most cancer research with niacin. Dr. Shuo Lin at UCLA, who taught me so much when I was a postdoc, and Dr. Taylor, who's just an incredible person, generous, my graduate school mentor, and Dr. Jarrett, one of the smartest people and community-minded, wonderful scientist, brilliant, who taught me that really the greatest scientists are the ones who ask the best questions. Yeah, thank you so much for your attention. So we can do questions now.
Okay, you can either unmute your speaker and ask the questions personally, or you can type the questions to Dr. Panberti straight away here. So the floor is open for questions.
Do I unmute?
Yeah, no, you are okay. I think we can still hear you. You don't have to unmute.
Okay.
Okay, I think there is a question in the chat if you check your chat.
Oh, okay. I have to open the chat window.
Yeah. You can see either at the top or the bottom.
Okay, I see it. Okay, what is the explanation for the loss of the ability to produce vitamin C in human? There's a gene called gulonolactone oxidase. All humans and guinea pigs and I think bats, we have a mutation in this gene, and it's a premature stop. That the gene is not fully made. We cannot unmute. Oh, okay, that's somebody else.
Yeah.
So we can't make it. There's some research with people that are small molecules that can revive the expression of this gene, supposedly, but that's very new. But we don't. But like a goat will make a ton of vitamin C when it's sick, but we can't. We don't, and we can really use it when we're sick. Okay. So do we need to measure niacin is another question. Is there a general recommendation? Okay. I don't see a need to measure niacin typically. As far as a general recommendation, the RDA is like 40 milligrams, but I think everybody could benefit from 3 times either 100 milligrams or 250 or 500. I personally take about 500 milligrams 3 times a day for 15 years. It varies. I buy the bulk powder. It's very cheap, and I can put that in a smoothie. But it depends on, you know, how much a person is suffering, I guess. But it's not a downside to it, really. There's unless you take really high doses, then you might worry about the macular cyst, or at least be aware of it. It's a rare thing. Okay, can niacin replace resveratrol for production of NAD, or do we need resveratrol as well? I think I like resveratrol. I've done experiments where I've compared things in zebrafish. I've compared that all of these things were compared because resveratrol and the NAD precursors work through sirtuins, and resveratrol is phenomenal. It was even more impressive than these NAD precursors. So I believe in resveratrol, and the mechanisms of it are just kind of all over the place. So I think it's good, and to take some resveratrol. Okay, which one is better, flush or no flush? Flush. Flush is better. So that's nice. Flush is associated with correcting all of lipodystrophy. It lowers your triglycerides if it's too high. It lowers your VLDL if it's too high. It lowers your LDL, your LPA if it's too high. It lowers C-reactive protein, and it raises the good cholesterol more than statins. The flush does that, the flush form, but not the non-flush. The non-flush is great too, and some people really can't stand the flush form. So they may have to take niacinamide. What is the maximum dose of glutamine? Would it cause brain fog? I have not heard of much adverse effects with glutamine. It is already the most abundant amino acid in your blood. And the clinical trials were 10 to 15 grams. So that's like about 3 heaping teaspoons of glutamine. And they got great results for burn patients. So it was oral, but then it was, you know, you can see the effects. So I just think you should, you can take it for any concerns and think of it like an NAD precursor. Really, yeah. In the context of someone battling cancer, though, glutamine becomes more complicated. It might not be advisable. You can look at Thomas Seyfried, S-E-Y, or contact me and we can talk about that more. Maximum dose of glutamate. I don't know about glutamate. It would be glutamine. Thank you. How can we reduce the flush? Oh, with the reducing the flush, it's just take less. It's just take, if you have to, do 50 milligrams. That's a lot more than nothing. I mean, sometimes something is infinitely, I mean, it's infinitely more than nothing. So you could do 50 milligrams or 100 milligrams, but a minimal flush is ideal. And then if you want to boost NAD for these people who cannot stand the flush, you can come in with niacinamide or nicotinamide riboside or nicotinamide mononucleotide or and glutamine. How about dose for high LPA? That would be, you know, 3 times 500 milligrams of niacin or 3 times 1 gram of niacin, typically. But again, you know, each person's individual. Can niacin replace? Okay, we got that. Should we be concerned with any other nutrient depletion? I'm not aware of other nutrient depletions, but there was that slight anemia in the muscle myopathy, and I don't know why that occurred. So I know that B vitamin imbalances can be a big concern, but with niacin, I don't understand. I don't know. entirely. It doesn't seem to be a common problem. How do we wean a patient off of benzodiazepines? I think it was 3 times 500 milligrams of either, it was 500 milligrams or 1 gram, 3 times a day of niacin or niacinamide. You can look up Bo Johnson's. It's in the, I think it was in one of those case studies I referenced in the previous slide. How to increase without flush the tolerance of niacin. Well, the other thing that helps, I should mention, is to take these things after eating. That can help. That can help. My wife likes it with yogurt. She said that helps her. Well, it decreases irritation in her stomach. Is it true that adrenochrome extracted from young children is used as a reverse aging protocol? I never heard that. I don't know. That's interesting. Is it okay?
Yes, can I ask a question?
Sure.
Yes, a short question. One of your slides shows that with the time release, you can get liver toxicity, while it said that even in higher doses, then it's relatively harmless. How can you explain this with the time release that is more harmful than the normal one?
I'm sorry, I couldn't understand which molecule are you talking about?
It's the NAD. The niacinamide. You said that's the NAD, and with a time release niacinamide, you can get a liver toxicity. If I'm not mistaken, it's one of your slides. Is that correct?
Lipotoxicity.
Yep. It was in your slide, if I'm not mistaken.
Maybe we could go to the slides and we can find the one.
That is one end of the liver toxicity.
This one?
No. Further on, talking on liver. It's only text.
Only text.
Yep. It's further on. I think it was only. It's only text. Further on, yeah, further. Just hang on.
This one?
Just hang on. Which one? No, further on. Talking on liver. On the liver.
This has liver toxicity.
Yeah. Just hang on. Liver concern is increased when saying only time release. This one, the second paragraph, the last.
Okay.
Only time release has had association with liver toxicity.
Yes. Yeah. I would have to get the review, but it's by John Guyton, and I can I can email you or something. He talks about this. They saw more of a risk with the timed release. I think somehow I don't know why that would be the case, but anyways, yeah.
Yeah, it's a bit to me confusing, because with time release, then you get a constant level in the blood, compared maybe the higher doses or by one-time administration. So there must be an explanation for that. I think. Is it due to the?
I think that maybe the clearance of niacin is meant to be some kind of a dynamic process that has to happen rapidly, and then things are changed. And then maybe if you keep delivering it with a timed release, somehow that's a problem. I don't know why.
Yeah.
I don't know.
Okay, thank you.
Hi, William. Such a nice presentations and quite challenging, I think. I'm Sofian from Jakarta, Indonesia. And my first question is, in one of your slides, you mentioned about David Sinclair, And coincidentally, this morning I read his book, Lifespan, the title of his book. And in his book, he mentioned, I quote, niacin and nicotinamide doesn't seem to raise NAD levels, as NMN, nicotinamide mononucleotide, and nicotinamide riboside do. So meaning that, according to his opinions, that niacin could be not really effective to raise NAD levels, as well as nicotinamide. What's your opinion on this one?
Who is that again?
David Sinclair, a geneticist from Harvard University.
Oh, David Sinclair.
Yeah, that's right.
Well, yeah, I'm very familiar. David has done just fantastic work, but that statement, I can't. If he said that, that's pretty shocking, because I'm sorry.
Yeah, the same with me, because actually I since 2 years ago I tried to daily intake niacin, and greatly helps me in increasing my HDL, and I feel that my mitochondria is quite good right now. But he mentioned this, as I quote just now. But anyway, okay, you can see on his book, Lifespan, on page 305. Okay, this is my second question. Could you please elaborate a bit the role of NAD as a cofactor? You mentioned in your presentation that NAD is a cofactor required to activate enzyme PARP1, which is needed for DNA repair. Okay, in terms of genomic stability. It's been presented in one of your slides, I think. Okay, but
yeah,
if you can explore it a bit, thanks.
Sure. Let's see. So
yeah,
for example, let me see if I can find a better one.
Yeah. No, that one. Yeah.
Okay. Yeah, it's.
I
have a better one. I really.
Yeah.
Okay. Yeah, that sounds good too. So whenever I have better picture tomorrow for this concept, but yeah, PARP is a really interesting enzyme. So interesting. There's been entire conferences focused on PARP. I mean, international PARP meetings that I've gone to focused on PARP. There's a PARP community, and it's a lot of it is Elaine and Myron Jacobson, and people all over the world. It's a fascinating enzyme. When you knock it out in a mouse, the cells don't die for a lot of stresses, because this pathway is devastating if it is activated too much. And a lot of radiation oncology works on this mechanism. It really causes DNA damage. Then PARP starts to use NAD and to make this what looks, it's a polymer, it's like NAD. It's a nucleotide polymer called poly-ADP ribose. And when the DNA damage is too much, or PARP is activated too much, so much of that NAD gets used up that the cell just collapses. It has no energy. It just bursts with necrosis, and you have collateral cellular damage. So PARP is, so the question is, what can we do here? And people have been developing PARP inhibitors to mimic that mouse, for example, because then, for example, you can almost blast a cell with radiation, and if you inhibit the PARP, the cell will live. As long as it's not too much, obviously. And it's similar for other stresses, associated with lots of oxidative stress that cause DNA damage and so on. So how do we approach this? Nick, you just try to, be ready for PARP activation. Multiple sclerosis, they've shown that inhibition of this will prevent exacerbation of multiple sclerosis attacks, for example. So, yeah.
Okay, thank you. My last question is, a couple of times you mentioned about mitophagy, okay, mitochondrial mitophagy, such as in the case of sarcopenia, okay. Will it be a good idea if I combine, let's say, NAD together with PQQ and also certain peptides, such as mitochondrial-derived peptides, as you may be aware, MOTSC, that can help directly in the process of mitochondrial biogenesis. I'm just curious, if we just use NAD solely, maybe the effect is not really effective. But if we combine with other compounds such as PQQ, quinone, and also certain peptides, maybe the result, the outcomes is more effective.
Yeah, I definitely agree with you. It's worth considering. I don't know, but I
Okay.
definitely subscribe to the belief that you shouldn't only do monotherapy if the situation allows for greater complexity, whether that's a smoothie or what.
Well, thank you so much.
Yeah, thank you.
There is some more questions on the chat group.
Okay. Would glutamine intake lead to increased glutamate? Oh, that's a good question. All I know is that from what I've seen, there's not an adverse effect. I don't recall any adverse problems with high doses of glutamine. Any experience treating cognitive disorder with these? This really needs to be done. There's been nutritional studies, definitely. Thiamine is huge with cognitive and Alzheimer's, but there just needs to be so much more done in this, and that's what my whole talk is about tomorrow. I would, and especially when you think about vascular dementia, which blood vessels, we can expect that 3 times 1 gram a day, or whatever's tolerable, or different combinations of the other NAD precursors should be expected to possibly be very beneficial. It's just a shame it hasn't been done. There's a mountain of evidence. It needs to be done. How long to see improvement? Mostly in schizophrenia. When to stop medicine? Say patient is on these. I think it can take a long time. I think Hoffer said it can take like more than a month for schizophrenics, and again, there's not really a downside. There's hardly any downside. Just taking high dose niacin make it not necessary to take NADH, or taking this would be useful with. You see, as far as taking like NAD or NADH, those molecules directly, they don't get into as many cells, and they do function as a substrate in the extracellular milieu and for different functions, but they're way more expensive, and I'm not aware that they're any better. I think maybe that acute stroke model shows that it may be in a critical care situation, maybe spraying it in someone's nose who's having a stroke may be life-saving. But I think generally the evolutionary, the evolved pathway is niacin more than anything. I think when some geneticists look at the bacteria and everything. What is the relationship niacin and NMN? Would taking high dose niacin increase NMN? And it is not necessary to take NMN. Okay, NMN is very interesting too. NMN may be the best. It may be, but it's just so much more expensive. And it's very much an area of active investigation.